Thanks, Scott. So I'm using both my hands here, one for the slide advance and one for the microphone. So please bear with me if this doesn't kind of work all the time. So let's try this first. Next slide, please. So I have no disclosures relevant to this presentation. And next slide, I would like to kind of give you one idea about why I'm actually giving this talk. Because technically, I could stop right here. I could say the genetics of Dravet syndrome is SCN1A and is due to loss of functions of SCN1A. And we could all kind of disperse, have a coffee, and go to the next talk. And this is probably what I would have told you in 2017. And next slide, please. So what I'm going to tell you now in 2022, it's actually the same. Dravet syndrome is due to loss of function variants in SCN1A. But there are many, many more things that I think you should know about. First of all, the rules by which we understand SCN1A, by which labs return results, are being changed. And this is a very big deal behind the scenes. Secondly, and this always sounds a bit paradoxical when I tell my trainees this, but genes don't matter. What matters are what genes do and what genes produce, so-called transcripts. And then finally, there's gain of function in SCN1A, and I'm going to talk about this, and I think our next speaker will kind of mention this too, that not everything that looks like Dravet is necessarily Dravet, and this is one of the key messages I want to relay. So here is my presentation. I'll talk about the history of Dravet, or to be more precise, the history of SCN1A. I will talk about uncertain significance in transcripts. So many of you have heard about variants and uncertain significance, that your doctor cannot say this is really a pathogenic variant, that this variant is uncertain. And I'll try to unpack this a little bit in a way so that you can understand this. And then finally, I'll talk about SCN1A beyond Dravet syndrome. So this is becoming a more and more important issue that I just want to touch up on. So, next slide. Sorry, and Veronica, I'm now on slide 6. Yeah? Let me talk about the history of Dravet syndrome and the gene that almost wasn't found. So let's go back to the early 1990s. The next slide, please. So here is one of our reviews from 2008, where we kind of try to summarize how doctors think about severe epilepsies in children, how they try to kind of put them into groups, and how to try to categorize them. And you can sum this up by saying Dravet syndrome belongs to a group of severe epilepsies of infancies for which classification has been challenging. You have 3 doctors, you have 5 opinions. Yeah? Which epilepsies are similar and which epilepsies are different. And this was actually a big discussion back then. So in Germany, my home country, Dr. Dose defined a disease that was called SIGA, the severe idiopathic generalized epilepsy of infancy. We don't talk about this anymore, but this is what he maintained is a good way to think about the epilepsies that now include Dravet. In France, we had Charlotte Dravet, who came up with the idea of severe myoclonic epilepsy of infancy, which is now named after her, which is Dravet syndrome. And in Japan, people talked about ICE-GTC, a intractable childhood epilepsy with generalized tonic-clonic seizures. Put all these people into a conference room, they won't stop talking. Yeah? So how can you solve this? You can imagine it's impossible if you try to understand now what belongs together, what is common. Yeah? But it got more complicated. So once people thought about Dravet syndrome, back in the 90s, there were quite some speculations of what Dravet syndrome could be due to. People thought about genetic causes, but for the wrong reasons. They thought, well, it's genetic because there's so many other family members in these families who have febrile seizures. We now know that this is not true, but at this time, this sounded convincing. On the other hand, there was an entire camp that says, well, it's probably acquired because we see so many seizures after vaccinations. Now we know it's the predisposition that comes with the SCN1A mutation that causes this, but back at the time, people didn't know. So we now know that Dravet syndrome is genetic, but the speculations were wrong. It's not due to family history, and it's not acquired. Yeah? And this is basically an entire shift in thinking that happened over the last 20 years. It's almost inconceivable for us right now what people thought back then. Just to give you an idea on how much we have actually done and how much the field has progressed. So, and I asked this question, I think, twice today. This person is Peter de Jonge. He's the guy who found SCN1A as the cause of Dravet syndrome back in 2001 in an environment that wasn't actually very conducive to gene discovery. And Peter wasn't really interested in looking at epilepsies. He just had one project where one of his students looked at this. And I forgot to say slide forward. I'm going to pause it, Veronica. I'm just at the slide right now where I have this kind of little Peter icon at the top. This is slide 10. So Peter kind of had this project going on where he just looked at this, and he didn't actually care what anyone else thought. Yeah? And he was one of my mentors back then, and this is kind of what I kind of took away from him. Stick to your guns, do your thing. Don't be so much concerned about what everybody else does. And he found SCN1A as the gene for Dravet syndrome. If it hadn't been for him, this would have been discovered 5 or 10 years later. And what we now think of as vaccine encephalopathy was demystified in 2006. So this is the Sam Berkovic and the Australian group. They looked back at a number of individuals who had an alleged vaccine-related epilepsy, and they found SCN1A mutations in a good proportion of them. So fast forward to 2022. Next slide. This is now an overview of the number of gene tests that have been performed in 3 major laboratories over a 4-year period. And you can see SCN1A is by far the most common result of gene findings. This is from our kind of paper that came off earlier this week on precision medicine. And SCN1A is really the most important gene in human epilepsy. So let me summarize this quickly for you. Dravet syndrome historically has been one of many concepts how we think about severe childhood epilepsies. Yeah? The etiology was very, very unclear before the discovery of SCN1A. SCN1A was found by the Antwerp team, and now it's the most common genetic cause in the epilepsies. And this is a story of how gene discovery helps us demystify things. It helps us kind of clarify preconceived notions. It's not due to family history, it is not due to vaccinations, but it is a genetic mutation. So what does SCN1A stand for? So this is next slide. Here is this is a nerve cell, this is a neuron. This is basically what makes us think, that is how our brains work, and we have billions of these nerve cells. And SCN1A sits here and is like the gatekeeper for all the electricity that runs down here. Yeah? There are other proteins, and SCN1A, as you know, is a channel. It's a little kind of pore that sits on the neuron. And this is what it stands for, SCN1A, sodium channel number 1 alpha. Yeah? This is what the SCN1A means. If there's SCN1A, there's also SCN2A. And you can see that the SCN1A gene, and I'm going to talk about what genes are in a minute, you can see that the SCN1A gene is one of many genes that have been linked to human disease. There's SCN2A, SCN3A, and SCN8A. Those are all the SCN genes that cause human epilepsy. So what exactly is a gene? What you can see here is a human cell. And on my next slide now, this is a human cell, this is a chromosome, and many of you may have seen this kind of cartoons. And then we take this chromosome and slowly unwind it. Yeah? This is what is called a karyotype. So this is a photo of our chromosomes. And the SCN1A gene sits right here in a place that we call 2q24.3. It's just the postal address of the SCN1A gene on chromosome 2. So from here, let's go a little bit deeper. This is the DNA. Again, we have the cell, the chromosome, and the double-stranded DNA. And this is now a chromosome that is stretched out. Yeah? And those are many, many different pieces of information, more than 100 million of base pairs that sit on this chromosome. And if you like to look at chromosomes, we do a so-called karyotype. That's the photo I showed you previously. Karyotypes are not that important in SCN1A and Dravet syndrome, but just for your information in case you hear about this, this is what it looks like. If we zoom in, and this is now a part of a chromosome, there can be little changes in chromosomes where a piece is missing or a piece is too much. And part of this may be the SCN1A gene, and this is typically assessed through a microarray. This is a kind of diagnostic test that we do in clinic. And then if we zoom in further, those are now the letters of the DNA. We go down to the individual base pairs, so G, A, T, C are the different variations, and here you can see a single gene and the single variant. And this is what we assess when we do a gene panel, an exome analysis, or genome sequencing. So both for families and providers, I have one important message. And Veronica, I think I missed my slides again, so I'm not going to know your GCP. Know your G dot, C dot, P dot. I repeat, know your G dot, C dot, P dot. So what does this mean, and why I'm telling you this? So this is the precise mutation that your patient or your loved one has. And it is very, very important to know this, and I'm going to tell you why in a minute. So G dot is the precise position on the genome. C dot is what is called the position on the RNA, the product that comes out of the gene, and P dot is the protein position. So G dot, C dot, P dot. You will find this on the testing report, and I typically recommend for all families to have your testing report somewhere. Typically when we have second opinions or when we see patients from other hospitals, the genetic reports typically don't automatically populate. The PDF files, and they're kind of stuck in each system's data bank. Yeah? So know your G dot, C dot, P dot. And why is this important? Well, this helps us understand if an SCN1A variant is truly causative. We all have changes in SCN1A, except that 90% of these changes don't matter. Those are common changes that half the world's population have. They're benign. And you could imagine it's that the way that these variants have been interpreted over time has actually changed. I'm going to give you two examples from our system here at CHOP. This is a kind of small epilepsy family, and this is what a family tree looks like. This is dad and mom and the two sons. So square is a male, circle is a female, and if the circle or if the square is filled, it means that these individuals have epilepsy. Here, this is a variant that was found almost 15 years ago, was classified as uncertain, filed away, and the test was forgotten. Because the doctors at this point thought, it's uncertain, it doesn't really matter. Yeah? Every time you have a variant in SCN1A that is called a variant of uncertain significance, look again. Every 2 years, ask your doctors, ask your providers, is this variant, what is new about this variant, is this variant causative? In this case, we dug out this old testing report. Yeah? This was a kind of a paper file somewhere, and found that this patient actually had been tested, and this variant is actually a causative variant. So the way that we understand variants in SCN1A changes over time, and it's always important if there's uncertainty to look again. Uncertainty also goes the other way. Here's a little boy who I saw in clinic last year who had an SCN1A variant who was diagnosed as Dravet syndrome. And we looked at this variant and said, well, let's see what we know about this variant. And we found this is a population variant, a variant that's been found in tens of thousands of people in the US. This cannot be a Dravet variant. We ruled this out, and this little boy actually had an epilepsy that could be treated with epilepsy surgery. So a false diagnosis had prevented him from getting the therapy that he actually needed. Yeah? This is why it's important to know your G dot, C dot, P dot. So we look at these families, and you can ask the question, why is there such a range here? Why does dad have febrile seizures? Why does one son have Dravet and the other one has GEF+, which is a mild SCN1A-related disease? In most cases, we don't know this, but here's one example from a very large family from Israel that we kind of published a few years ago. And what this family actually has, it has two SCN1A variant, one that causes Dravet syndrome and the other one that prevents Dravet syndrome. So some of this variation in families can sometimes be explained by what is called modifier genes. We know there is a wide range in SCN1A, and part of this might actually be explained by other changes in this gene. So I was asked to talk about the genetics of Dravet syndrome, and I got stuck on one single gene, SCN1A. What about all the others? So several different genes have been reported as Dravet mimics. And Veronica, I'm not on this slide. What about other Dravets? And this is slide 22 in my deck. GABAG2, PCDH19, CHD2, KCNA2, HCN1. I don't want you to remember these genes because those are not Dravet syndrome genes. They sometimes have some similarities in the early stages with Dravet syndrome, but in virtually all cases, the clinical presentation is much, much different afterwards. So we think of them as mimickers, and here is a slide that Andreas Brunkhaus gave me today from his publication. Dravet syndrome basically equals SCN1A. And this is even true if your patient or if your loved one is negative for SCN1A. So the most likely gene in SCN1A negative Dravet syndrome in individuals with Dravet syndrome who do not have SCN1A mutation is SCN1A. When you look again and do another test, you're more likely to find a missed SCN1A mutation than any other gene. And we kind of actually looked at this, and we spent quite a bit of money looking for other genes for Dravet syndrome just to find SCN1A again. Yeah? So there's a big kind of project here from our initial work in Europe. So Dravet syndrome is SCN1A. There are a few other genes that mimic Dravet syndrome, but this is not something that is very kind of important when we think about a Dravet. So let me talk about uncertain significance in transcripts. From these two examples I showed you previously, you already got a sense that it's actually quite important to understand if an SCN1A variant causes Dravet syndrome. And typically we think about this as a circle. We have our patient here, we want genetic testing, this testing gets results in a report, and this is now being fed back to the clinic. Typically we think about these two parts, doing the test as quickly as possible, and then returning the results as quickly as possible. What we typically don't think about is what happens behind the scenes. How do we go from the testing report, or from the actual test, to the testing report? Yeah, and this is where what is called a gene curation. This means how do we interpret a genetic test and what goes on the report. This is what the lab does, and this is what happens behind the scenes. Right now, the rules for SCN1A are very generic. When labs look at SCN1A, they use the same rules that they would use for breast cancer genes or for genes for hyperlipidemia. Yeah? And this is why we have so many variants of uncertain significance. We're part of a working group that you probably don't know about, which is called ClinGen, and we're officially tasked with rewriting these rules that are going to be used by all diagnostic labs throughout the world. This is our ClinGen Epilepsy Working Group. It's headed by Heather Mefford and me, and we're basically kind of tasked with rewriting the rules. And as this work happens largely behind the scenes, I just wanted to quickly give them a shout out because there's quite a bit of thought and quite a bit of work of aligning this. Yeah? The reason is the following. When we look at how a lab interprets SCN1A, they do this by very generic rules, and sometimes rules that are more than a decade old. For example, here, in the official lab reports, until early last year, Dravet syndrome was called an early infantile epileptic encephalopathy. So Dravet syndrome starts in infancy, but not really neonatally, not really kind of in the first few months, and I'll come back to this later. And so one of the things that we did already is we asked OMIM, which is this big dictionary that everybody uses, to actually do away with this classification, and as of last year, they're not using this term EIEE anymore, but using Dravet syndrome. Second point. We need to start thinking about not the actual gene, but what the gene produces. SCN1A is a gene. It is a gene on the second chromosome, but there are many different ways how our body extracts information from this chromosome. And you've seen this yesterday with some of these presentations about antisense transcripts. So there's not just a single SCN1A product. There are multiple SCN1A products. There are SCN1A products that actually inhibit the true SCN1A. And this is what all these antisense therapies are aimed at. They're trying to kind of do gene regulation using the naturally occurring changes in SCN1A. Here's one example that Ethan Goldberg and I kind of published as a commentary last year. We know there's the so-called poison exon, which keeps SCN1A expression down until the age of 6 months. And this is when SCN1A is needed, and this is typically when Dravet syndrome starts. So we need to think about more than just the gene structure, but what the gene actually does and what the transcripts are. So here's one reason why this is important. I'm going to come to my last point now. When we think about SCN1A in a broader context, and this is, I think, one message that is very timely to send right now, because there are many publications coming out right now that we've probably called quite a few conditions Dravet syndrome that are actually not Dravet syndrome. And when we think about what the mechanism of typical Dravet syndrome is, it is the loss of function, either the loss or a stop mutation of a single copy of the SCN1A gene. However, we also know that there are mutations, changes in SCN1A, that make SCN1A more active. Here's one early example. This is the first genetic change. This is the change, it's the P dot, G dot, C dot, P dot. This is the P dot here of an SCN1A mutation that leads to DEE, which is developmental and epileptic encephalopathy, a very severe neonatal epilepsy with movement disorder that is also due to mutations in SCN1A, but is not Dravet syndrome. So there's a subset of SCN1A-related changes that are not loss of function, but that are gain of functions, where the SCN1A gene is too active. And until 5 years ago, we didn't actually know this. And there are probably quite a few patients misdiagnosed as Dravet who have a gain of function variant. So can we kind of be a bit more precise in what we are looking at, and can we kind of estimate this? We've tried to. Here, it's just an overview of our entire healthcare system when it comes to Dravet syndrome. This is patient age. Here are Dravet patients stacked by the earliest encounter in a healthcare system. Every dot is a single patient encounter, and what you're looking at here is roughly 100,000 patient encounters in more than 200 individuals with Dravet syndrome at Children's Hospital of Philadelphia. This is basically data science. This is what my lab does. We try to extract this information from the medical records. And now we've done the following. We've arranged our patients with Dravet syndrome by the age of onset of their epilepsy. This is the typical range. This is 6 to 9 months when we think Dravet syndrome typically starts. But you can see here, there are quite a few individuals with SCN1A mutations who have seizure onset much, much earlier as the first day of life. And what was interesting to us is when we look at the mutations here, we found the very same mutation in 4 individuals who had this very early onset. This is basically 4 individuals out of 2,500 patients we've seen in the last few years for epilepsy genetic workups. It's the most common genetic change in an ion channel that we've seen. This is almost paradox. If you had told me this a year ago, that the most common change in an ion channel gene is an SCN1A variant, I would not have believed you. But this is an SCN1A variant. We call this the Philadelphia variant. This is a gain of function SCN1A variant. And what this tells you is that there are gain of function variants. And if you want to think about what our patients, what our kind of family members have, it's important to go back and say, when did this actually start? And what I'm challenging everyone, if you have a patient or a family member who has had epilepsy that started way earlier than 3 months, revisit this again. It might not be a loss of function, it might be a gain of function variant. So, SCN1A beyond Dravet syndrome. Andreas is going to talk next, has just kind of published a very nice paper on the SCN1A gain-of-function spectrum. This is increasingly recognized. It is the kind of the anti-Dravet, if you like to think of it this way. And this is not limited to kind of very severe syndromes, but this may also be one of the mimickers, one of the masqueraders of Dravet syndrome. It's under-recognized, and age of onset is critical. Again, any SCN1A disease that starts before 3 months is suspicious, not of Dravet, but of an SCN1A gain of function. And this is important because many of the medications that you've heard about earlier, sodium channel blockers, they may actually be useful, and they may be even leading to kind of treatment and seizure freedom in these patients. So I try to give you a 2022 update on the genetics of Dravet syndrome. And I talked about the history, the gene that almost wasn't found. So SCN1A was found against all odds by Peter de Jonge. I've talked about how we're trying to kind of be more precise about SCN1A diagnostics, trying to improve the rules to make diagnoses faster and to eliminate much of the uncertainty that comes with testing reports. And then finally, I've given you an idea about SCN1A gain of function. Again, you can see, and Veronica, I'm on my second to last slide right now. Sorry, I kind of missed this the last 2 minutes. There's much to say about the genetics of SCN1A and the genetics of Dravet syndrome. Yeah? It is not just the fact that SCN1A is due to loss of function variants. We're changing the rules. We're looking at other transcripts, which are now being used for gene regulation therapies. And there's basically more to SCN1A with severe epilepsies than Dravet syndrome. So I'd like to finish by thanking my lab. I'm happy to take questions. I think Scott is just finding the second microphone. Otherwise, I can just walk towards you. That's fine. Why don't you speak so that everybody else can hear this too?
Can you hear me now? Okay. So, very interesting discovery about the gain of function mutations. What is the way to distinguish between the scenario of having a gain of function mutation versus mild loss of function mutation that is enhanced by a modifier in the genome? How do you go about that? And the second question is, has there been a systematic analysis of protective variants in other protein coding genes? Because as far as I have seen, most of them are enhancing, right, the risk, the burden. So, thanks.
I think it's okay if I stand here, right? Then I don't need to go back and forth all the time. So the first question was, how do we kind of think about, how can we distinguish gain of function from, let's say, milder Dravet cases that might have been kind of modified by genes? And the answer is age of onset. So even if we have a milder Dravet presentation, this typically would not start before the age of 3 months. So again, I think the rule of thumb that we're trying to establish right now is every SCN1A-related epilepsy before the age of 3 months, definitely before the age of 2 months, is very suspicious of a gain-of-function mechanism. And this is important because the treatment might be completely different. For your second question about modifier genes, so I come from a clinical genetic background where we have a very high threshold for calling genes true modifiers. The one example I showed you with this large Israeli family is really just kind of a single explanation of how this might work. So far, there's no systematic analysis out there. There's some interesting ideas from mouse models, but there's nothing right now clinically actionable that we can test in humans. In most cases, this is going to be due to hundreds of different genetic changes that we all have. We know this from common epilepsies, that those are many hundreds of different kind of common changes. And this probably also explains some of the changes that we see between our individuals with Dravet syndrome. There was a question here, yeah.
Our son is 41 years old now, and he wasn't tested till he was 30 years old. Back then, I have no idea what this G.C.P is. Was he tested for that, or was it 11 years ago? I was wondering how advanced the testing was.
So this is a very good question. So I implored you all to think about G.C.P. And what this basically means is find the old testing report. It's going to be in there. And show this again to your geneticist or to your doctor and say, now in 2022, what do you think? Yeah, and I think this is, in most cases, these tests are right, but if they're wrong in 1 in 10, it's important for us to actually know. I had another question, I think, on this table here.
Hi, I'm Ron Cantor. I'm actually a cardiologist, electrophysiologist, and we've been in the cardiac channelopathy business a very long time. And as such, we've been really tasked with having to look at what was once considered pathogenic gene mutations, and now it's being rethought. And what we're finding is that over the years, they're being reclassified and more often going from what was once thought to be probable or definite pathogenic to more likely not pathogenic, emphasizing the importance of a proper phenotype and the clinical presentation. So my question is, what do you anticipate it going also in a direction of down classification, up classification, or are you guys getting it just right?
All right, so this is cardiology throwing some shade here, yeah? So the idea is the following. So there's probably 10 or 100 times more genetic testing being done in cardiology than in epilepsy. Epilepsy is always one step behind, 2 steps behind, which makes me mad. This is why we're here, yeah? So what has happened in the cardio...
Proportionate to the importance of the organ, believe me.
All right, I'll come back to this later. We'll take this offline. Brain always wins. Yeah. So the idea is that the following: that genetic tests change over time, and I think your question was what we see in the cardiac field is that many changes that people used to kind of call disease-causing 5 years ago are now not disease-causing anymore. Not because the gene changed, not because this genetic change has mutated, but because our understanding of what this change does is now different. Now we've seen that a change that you might have seen 5 years ago or 10 years ago in a patient and said, oh, this must be causative because there's no one else out there who has this, therefore it must be causative. Now we know that half the population of Fort Worth has this kind of change. They don't have cardiac disease, therefore this change is very likely not causative. So I think the advantage that we have in the epilepsy field is that we know all this now. Yes, I think the idea on how precise we are with understanding our variants is actually informed by this already. So all the rules that have been found to be true in the cardiac field and the cancer field, we already know about. And this is why I think we have don't have so much back and forth. So we often take away diagnoses. So I typically tell families a third of my job is actually undiagnosing genetic epilepsy because there are many false positive changes out there. Dravet syndrome is, of course, underdiagnosed, but it's also well known. So people sometimes tend to stick the label of Dravet syndrome to diseases that are not really kind of adequate. And at the same time, especially as in many kind of older individuals, they may have had testing. This testing may not have kind of shown an SCM1A change, but it's actually there. So I think it goes both ways. I think there was one more question. You guys make me walk through the entire room here.
Thank you. So I'm a mom without any medical training, so I need to ask you, what do you mean know your C.G.A. I know what is his C. But what does it mean in reality? Can you predict the severity of the Dravet or?
Yeah. let me take one step back. So when Veronica and Scott asked me to give this talk, they kind of gave me an impossible task. And so this should have been a presentation for families and for doctors. And let me maybe kind of clarify this because I should have explained this better, so I apologize. Yeah? So what I mean by know your G.C.P. is means have this report available somewhere. Yeah? Don't rely on a patient letter that says your child has Dravet and this was tested at some point ago, but we don't have this test report in anymore. Have this there and just make sure that your providers can look at this. Yeah, and sometimes what we even do is we kind of talk to families about this during their visit. We say, here's what these things mean. This is where the mutation is actually on the SCN11A gene. Probably takes your doctor 5 minutes. So sometimes it makes sense to pull out this testing report again and say, please explain this to me. And then often things are clear. When there's any kind of uncertainty, we can go back and say, let's look at this again. Maybe there's some kind of question mark there. Maybe it's not really a Dravet gene, or maybe there's something more to it. This is what I meant. Just have the testing report somewhere that your doctors actually can look at this and kind of challenge them to go over this with you.
As our understanding has evolved, has it changed the view as to whether it's hereditary or not?
That is a very good question, because Dravet syndrome is genetic, but in many, many cases not hereditary. So I'll give you a different example. When you think about Down syndrome, an example that probably makes many people know, Down syndrome is due to an extra chromosome, and it is a genetic change because there's an extra chromosome, but it is not hereditary. It is not kind of inherited from families. Sometimes people mix this up, and this sometimes happens even in like newspapers, that they mix up genetics and hereditary. But the vast majority of severe genetic epilepsies that we know, they are due to what is called de novo changes. So changes that are in the patient, but not present in the parents. And this is the most common mechanism in Dravet syndrome. I should have also put this into my presentation. I apologize, but thanks for asking this.
Okay, we're going to make this the last question.
I have 3 more.
He is here all weekend.
I did want to follow up on the other mom's question that she asked. I'm also a mom of a child, and so you explained, know your G.C.P. But I guess I'm really asking, from a parent perspective, if you have a report that shows you have the SCN1A gene, knowing that additional information, what's the value of that in terms of our clinically and understanding the impact on our child? Does it mean that they are potentially, you can see from the more detail that they're going to have seizures, certain things, or they're going to have gait issues, or they're going to have, is there a reason why you feel that additional information is going to be helpful in terms of what we need to know about our child?
So I've accomplished one thing. I made you guys think. This is good. So what I meant by this is that this is information it's important to have, first of all, to revisit this for certainty. And I know because in many cases we don't really have this. And this is basically my clinical experience, that I'm often seeing patients with Dravet, and then someone has done a test report at some point where we don't have this. And it's good to have this. This will probably be important in the future when it comes to clinical trials, where we need this very precise information. And it's important to kind of see, is this really a true Dravet, or is there something different about this that we know from the genetics itself? Scott, I had 2 more questions, and then you can cut me off. Yeah? So I had 2 more questions here. Who was first? Thank you. Yeah.
Thank you. My first question, Dr. Perry, what's Katie C. or P. mean? I'm just kidding.
Yeah, no, I've got them all memorized because that's what I do.
No, my real question is, have you, is there any correlation in your research on where the mutation falls on the gene, like in the beginning, middle, end?
Thank you. So the question was, does this kind of G.C.P. play any role when we think about what the mutation is? And let me take maybe a step backwards. So this is what is called a genotype-phenotype correlation. So in individuals with Dravet syndrome, is there actually a difference whether they have a stop mutation at position 1 or at position 500, or if the entire gene is deleted. In most cases, not. So in most cases, we think that the mutations that cause typical Dravet syndrome, they all lead to one copy of SCN1A not working properly. Then it doesn't really matter where in the gene this mutation is. Sometimes we think milder SCN1A diseases like GEFS+, these changes might be different. Yeah, these changes might not lead to a complete loss of function, but might only kind of change the gene a little bit. But for 90% of Dravet, it does not matter. And then, yeah.
How does this apply to gene deletions?
You guys really know how to ask follow-up questions. Very good. So a gene deletion is the following. So you could imagine that there is a change in the SCN1A gene that is not just a change as a mutation, not just a single change, but that the entire SCN1A gene is missing, or that part of the SCN1A gene is missing. If this is only the SCN1A gene itself, it is the same on the genetic level or on the transcript level. Remember, we don't care so much about genes, we care about what genes actually do. On the level of what is being produced from the chromosomes, it's the same as if you had a stop mutation or if you had a different mutation. This is important because often when we have families and they kind of come back to us and say, my child has a deletion and not just a stop mutation, they would think that this is more severe, but it's actually not. So it doesn't really matter in terms of what the gene does if you have a mutation in the gene or if the entire gene is missing on one copy.
Great. Thank you. Thank you.